Doesn't jump up.
Hello everyone, welcome to Homeopathy Study Group Pro Bono webinar. Good morning, good afternoon and good evening. So nice to see many of you from different countries and different time zones. Before we start our webinar, always thank universe for giving us this great opportunity. And Dr. Sweta Singh, our Chief Administrator, will start the session.
Hello everyone, this is Dr. Shweta Singh, Ka Chief Administrator. Welcome you all in Ka Homeopathy Study Group Pro Bono webinar. Let me introduce Ka Homeopathy Study Group with all of you. Ka Homeopathy Study Group Pro Bono was organized and founded by homeopath and humanitarian Kavita Kukunur, President and CEO of Kavita Holistic Approach, Ka. This study group is intended to be an offering from Kavita to the homeopaths around the globe, sharing goodwill and solid clinical work within the classical model are foundational principles for the Ka mission and vision are very unique to inspire young homeopaths, mentor, provide excellency for educational purposes using holistic approaches via webinars, which are based on principles of classical homeopathy and provide professional continuing educational homeopathic credits for practitioners. We provide merit certificates for spreading the light of homeopathy around the globe. Dr. Kavita is a member of Kevin Friendly Foundation, a non-profit organization that helps to serve poor people in greater needs in India. She is the recipient of Martha Allman Community Service Award by National Center for Homeopathy and Best Entrepreneur Award from Dr. N. Linga Raju, Principal of JSPS Homeopathic Medical College, Hyderabad, India. We are extremely happy and proud that we celebrated 10 years of her book, Beyond the Limits, a challenge to prove oneself, and we have happily celebrated her second write-up, the ebook, Dose of Spirituality with Kavita. Dr. Kavita donated the proceedings of the book through Ka Study Group platform to various charitable organizations and homeopathic associations. To name a few are Kevin Friendly Foundation, CHC, DHMA, JSPS College, VT Seva, NCH, etc. As of now, we have over 250 plus recordings on professional webinars related to homeopathy, health related topics, and inspirational talks available on our channel, Kavita Kukunur. Please note that these webinars are for educational purposes only and see an expert homeopath for treatment. Any webinar content should not be used without speaker's permission. The webinar is being recorded as we speak, and we will take questions at the end of the webinar. We will post jot form in Zoom webinar chat at the end of the webinar. Please fill the form to receive certificate, and kindly mute yourself and turn your video off for better connections. We have with us our coordinator, Dr. Shrija Yeravalli, and she will read the code of ethics. Over to you, Dr. Shrija.
Hello, ma'am. Thank you so much, ma'am. It's good to see you all today. And I'm going to read the Ka Homeopathy Study Group Pro Bono Ethics and Benefits. To be a Ka speaker needs at least 1 to 2 months active volunteering. Only Ka speakers' teams are promoted in newsletters and webinars. Ka provides volunteering for 2 to 12 months, respects and inspires volunteers to grow. Active volunteers will be promoted through our website, social media, and added to WhatsApp group. Articles of volunteers will be posted on KHA website and newsletter and will be sent for publication at and other journals too. Volunteers and speakers are eligible for prestigious KHA annual awards. Volunteers are eligible for KHA inspirational talks. Volunteers are eligible for case discussions. KHA promotes homeopathic organizations as well as their mega events. KHA promotes great work of speakers through their book reviews. Honorable guests may propose their future courses or events after the webinar. KHA maintains a healthy and happy environment, so keeps only like-minded folks. Thank you so much, ma'am. Over to you, ma'am.
Thank you, Dr. Srija, and she will be moderating today's webinar, and she will take the questions at the end of the session. Today's topic is very interesting, experimental homeopathy. A need of the hour is research-based homeopathy, which is a great challenge for homeopaths. Today, we will learn more from our honorable guest speaker, who is an eminent homeopath, Dr. Girish Gupta, with over 40 years of experience in his clinical practice. He got BHMS degree in 1981 from Neminath Homeopathic Medical College, Lucknow. He worked at Central Drug Research Institute as research scholar in a project entitled Screening of the Homeopathic Drugs Against Animals and Humans Viruses, sponsored by CCRH from 1982 to 1985. He did MD Homeopathy in 2008 and PhD in 2016 from Homeopathy University, Jaipur. He was principal investigator in 3 clinical research projects, role of homeopathic drugs in PPH and ovarian cyst, sponsored by CCRH, and PCOD by Ministry of Ayush, Government of India. He started his private practice at Lucknow in 1982 and established Gurang Clinic and Center for Homeopathic Research in 1995. He's the president of Homeopathic Research Foundation, that is HRF, and Asian Homeopathic Medical League, AHML, an international organization. He has published about 70 research papers in various journals and 3 books entitled Evidence-Based Research of Homeopathy in Dermatology and Gynecology. The latest book named Experimental Homeopathy, which was released in this April 2022 in the Scientific Convention on World Homeopathy Day. Many congratulations, Dr. Girish Gupta, sir, for release of your new book. And let us welcome Dr. Girish Gupta, sir, to our show. Welcome, sir.
Thank you. Thank you, Dr. Shweta.
Thank you, sir.
Thank you, Dr. Shweta.
Yes, sir. You can proceed with your session, sir.
Okay. So thank you very much for your very exhaustive and comprehensive introduction. One correction I would like to make.
Yes,
I
sir.
passed BHMS from National Homeopathic Medical
National.
College and Hospital,
Yes.
Lucknow. At
Okay.
that time, Neminath College was not in existence. Rest
Right.
everything is fine. Rest everything is fine. So let me first of all, thank Kavita Holistic Approach Group in general, and Dr. Kavita Kokunur, Dr. Shweta, and Dr. Srija Yaravali for conducting this webinar and planning this webinar. This is indeed a proud privilege for me to be in front of you to show you totally different kind of research work in homeopathy. Instead of clinical, it's experimental. So I would like to tell you, when I was doing BHMS, I was in final year. Then what happened? A news article by a professor of medicine in King George Medical College appeared in newspaper, telling that homeopathy is a placebo therapy. This is a psychotherapy. So this was a very big jolt at that time. So I decided to show to the world that it's not placebo, but it's a medicine. It's biologically active drug. So what I thought that let our drugs, potentized drugs, be experimented on plant diseases, where there is no nervous system, there is no psychology. And objectively, I could be able to show that these drugs are potent one, they are biologically active and not placebo. So this is how the things started. So I contacted several scientists of National Botanical Research Institute based in Lucknow. And after great persuasion, I was able to plan some experiment, design some experiment over there. So the first experiment we did was on tobacco mosaic virus. And you'll be surprised to know that very positive results were obtained after testing homeopathic drugs against tobacco mosaic virus on plant. So that encouraged all of us to work further. And this was enough to show to the world, scientific world, that these drugs are active even against plant viruses. So this is how we started working. So our topic now today is experimental homeopathy. That is based on my book, which was recently released on 9th April 2022 during a World Homeopathic Congress organized by Central Council for Research in Homeopathy in New Delhi. Dr. Sweta was a witness, eyewitness to that release process. So slides are visible to all of you, Dr. Sweta?
Yes, sir.
Okay. So this kind of research was conducted at different institutes, namely Department of Plant Pathology, National Botanical Research Institute, Lucknow, followed by the other work on animal viruses in the Department of Virology, Central Drug Research Institute, Lucknow, and National Research Laboratory for Conservation, Lucknow. These all three institutes are under central government, that is Government of India. The two institutes, the number 1 and number 2, are working under CSIR, that is Council for Scientific and Industrial Research under Government of India. And the third one is under Department of Science and Technology, Government of India. And the last work was done in Medical Mycology Lab of Gaurang Clinic and Center for Homeopathic Research. We ourselves established later on in our Gaurang Clinic Medical Mycology Lab in collaboration with the medical mycologist, Professor Chandra and Dr. Srivastava. This is situated in Lucknow itself. This is the premises of Gaurang Clinic and Center for Homeopathic Research, in which number of OPDs and that medical mycology lab and pathology lab is situated. This is just a glimpse of medical mycology lab of our Gaurang Clinic and Center for Homeopathic Research, in which a number of experiments have been conducted in collaboration with the scientists. I would like to start with the inhibitory activity of homeopathic medicines against human pathogenic fungi, an in vitro study. Because as we know that homeopathy is a subjective science. A drug is given to patient and the symptoms are recorded. Whether the drug, if drug has acted, you find amelioration in symptoms or even cure. Otherwise, if it doesn't work, then you don't get the result. But it is very difficult to objectively show as to how homeopathic drugs are working. So this is how an in vitro study was planned long back in 80s. Now the research question is, that homeopathy has always been termed placebo. Even today, even these days, people usually say it is placebo and psychotherapy. It is criticized by conventional physicians and scientists as well as not being an evidence-based medicine. This is, I mean, the big question in front of homeopathic doctors to prove it otherwise, to prove that homeopathic drugs, homeopathic science is evidence-based. So the research design was uh, to it was difficult to prove biological activity of potentized medicines on human beings because you cannot show. You just prescribe the drug and you can see the betterment in symptoms in pathology, but you cannot objectively show to the world that these drugs are working. That is how an in vitro experiment was planned to achieve objective response that can be seen by eyes or by microscope. So the main objective was to prove that homeopathic potencies are not placebo. This was a big question in front of us, and in particular to elicit antifungal activity of potentized medicines in vitro model. We earlier worked on viruses also in Central Drug Research Institute and in NBRI against plant viruses. But here we are restricting our presentation on antifungal activity of homeopathic drugs in vitro model. Let me clearly tell you, our main aim was not to prove any activity against fungi or to find out any antifungal drug, but my basic purpose of conducting experiment was to prove that homeopathic drugs work. They have got biological potential to inhibit the pathogens in vitro models. Various homeopathic drugs and potencies were tested against different species of human pathogenic fungi using different experimental protocols. And a standard antifungal drug, that is ketoconazole, rectified spirit, and distilled water were kept as control because you have to have a controlled study. In one side, homeopathic drugs are there. On other hand, rectified spirit as a negative control and ketoconazole as a positive control. And distilled water also was kept as a control just to show the difference of activity of our drugs. Fortunately, the results showed that potentized homeopathic drugs had definite activity, inhibitory activity against fungi, confirming that these medicines not only work in vivo, that is on the body, human system, but are equally effective in vitro conditions. For any research study, you have to have some materials, methods. So we worked on several fungi, namely Candida albicans, Aspergillus flavus, Aspergillus niger. Aspergillus niger is a black fungus. Candida albicans is a white fungus. This was in context with the post-COVID complications infected with these fungi. So it became very relevant during Corona times. The fourth fungi was Carvularia lunata. Fifth was Trichophyton mentacrophyte, and the sixth was Microsporum adulani. So these are all human pathogens. Aspergillus niger and flavus causes fungal infection on the skin as well as in lungs. Trichophyton is a dermatophyte. Microsporum affects your hair follicles, leading to hair loss. So these all fungi were isolated from patients of Goran Clinic, those who were suffering from infection in the hair, nail, or skin. These isolates were taken out from the patient directly by the method Rona M. Mackey. This is a internationally acceptable process of isolation of fungi from patients. The other materials used were distilled water, rectified spirit, ketoconazole, and the quantity of drug was taken as per their MIC calculated. Experimental protocol was used as a disc method. There are several strip method, disc method, well method. So here we used disc method. How we assessed was measurement of zone inhibition. If there is any drug activity, then there is a inhibition zone. And the second was viable colony count. The third was inhibition of linear diametric growth and sporulation. If there was any result or effect of homeopathic drugs, these were assessed by all these 4 parameters. Now coming to show you how we conducted the experiments. These are culture plates. There are 9 culture plates. The first one was controlled. It has got Candida albicans grown under experimental conditions. The other one is positive control, that is ketoconazole. You will see a zone of inhibition around that disc that shows that ketoconazole is effective against Candida albicans. Now look at homeopathic drugs. The third slide shows, the third plate shows activity of Meserium 200. The next one is kali iode 30, acid benzoic 30, petroleum 200, sulfur iode 30, sulfur 200, and tellurium. You will see that if you compare the zone of inhibition with that of ketoconazole, Meserium 200 has given the best inhibition against Candida albicans. This technique is known as inhibition zone technique, in which rectified spirit has given only 12 millimeter, whereas ketoconazole has given 35 millimeter zone of inhibition. Compared to that, Meserium 200 has given the best response, then followed by other medicines. So that shows our Meserium 200 is effective against this fungus compared to the control, whereas rectified spirit is a placebo. It is not working at all. So how Meserium is different from rectified spirit is visible from this kind of experiment. This was a very comprehensive study. It was published in a biological memoir. This is not a homeopathic journal, but it's an international journal published by our International Society of Applied Biology. This was published in December 2003 issue under the title In Vitro Inhibitory Activity of Some Homeopathic Medicines, Drugs Against Candida albicans. So this was highly applauded in the year 2003. So we were encouraged to do further work. Now this slide will show you inhibition zone technique. In all the drugs are here taken in 30 potency. If you compare distilled water, it has given 0 inhibition. Then rectified spirit has given 0 inhibition in plate number 2. And the plate number 3 is treated with ketoconazole. You see a very large zone of inhibition, that is about 40 millimeter. And if you compare with others, acid benzoic, it has not given any inhibition, followed by apis, graphitis, kali iode. But here you see Meserium 30 has given some zone of inhibition. Other drugs are not effective. So you can objectively show to the world that if all the drugs are placebo, then how could only Meserium work, others are not working? So this is an objective model to show that our drugs, all the drugs are quite different in their activity. This is a different slide. These drugs have been tested in 200 potencies, Apis mellifica, and here you will see the Meserium 200 has given much better response compared to acid benz, apis, graphitis, sulfur, silicea, sepia. These drugs are not effective at all except Meserium 200. which can be compared with ketoconazole. If you look at the plate number 3 on the top and compare with 8 number disc, that shows meserium 200 has given a very, very good zone of inhibition. So this is how you can see, I mean, even a blind person can appreciate the activity of homeopathic drug. If it is working, otherwise not. So in the same way, this is a graphical presentation in which we have compared the activity of ketoconazole and other homeopathic medicine. So if you look at the blue line, meserium 30 and 200 has given better response compared to acid benzoic, apis, graphitis, petroleum, sepia, salicia, etc. Sulfur 30-200 has given some response, but meserium came out to be the best drug. I will show you later on how we have used meserium in conducting further experiments to test whether edible items have got any antidotal activity on homeopathic drugs. I'll show you later on. So fortunately, this very experiment was very successful, and it was published in the peer-reviewed journal, Indian Journal of Research in Homeopathy, published by Central Council for Research in Homeopathy, Ministry of Ayush, under the title Anti-Candidal Activity of Homeopathic Drugs and In Vitro Evaluation. It was published in April-June issue, 2015 issue of IJRH. You will be surprised to know that this paper was revised 9 times by the reviewers. Then only we could convince them, we could, get through the publication, green signal for publication of this article. So this was a big, encouragement to me and my team that this work was published in a peer-reviewed journal. Now I am showing you one drug and against Aspergillus flavus. Here only one drug has been taken in different potencies, just to cross-check. Thuja Q, Thuja 30, Thuja 200, Thuja 1M, Thuja 10M, Thuja 50M. These drugs have been tested against one single fungus, that is Aspergillus flavus, and you will see in control it is sterile water. Then number 2 slide, plate is treated with rectified spirit, and positive control is treated with ketoconazole, in which you will see almost 100% inhibition is there. If you compare with different potencies of Thuja, you will see the best inhibition zone has been made by Thuja in 30 potency and 200 potency. Other drugs like, in different other potencies, Thuja has not worked. So here only 30 and 200 potency is effective, meaning thereby our potencies act as a different individual drug. Here drug is the same, potencies are different. You will see 30 and 200 has given the better response compared to 1M, 10M, and 50M. So these drugs are, these potencies are per se, these are drugs themselves. One potency is different from other, which is visible even to the naked eye. This slide shows you the same graphical presentation. The green globule is ketoconazole, and Thuja 30 and 200 has given better response compared to other potencies of Thuja. So this is quite obvious that our potencies are quite different from each other. Here, the same experiment has been repeated with another species, Aspergillus niger. This is a black fungus. This is a black fungus. And you will be glad to know when there was a menace of secondary infection with the black fungus during post-COVID time, last year in 2021. Then I submitted this work to the Ministry of Ayush, to the secretary, who made a very good team of 18 doctors across the country. And on the basis of this work, which I did in 1995, an advisory of, advisory was issued by Ministry of Ayush to use, to procure, to utilize effect of homeopathic drugs in post-COVID infections infected with black fungus. This is Aspergillus niger. Fortunately, Corona came into 2020 and 2021. This work was published way back in 1995 by me and our team. So here you will see, if you look at the black fungus, plate number 1, this is a black fungus. Totally, it is treated with sterile water, meaning thereby water has no effect on fungus. The other is treated with rectified spirit. There is some inhibition. And the third one is treated with ketoconazole, which is a known antifungal drug. It has given a very, very good inhibition. If you compare with the different potencies of Thuja, here against Aspergillus niger, you will see Thuja 50M and Thuja 200 has given better result compared to Thuja 30, Q and 1M and 10M. So you just see the effect of Thuja in different potencies on 2 species of Aspergillus. One was Aspergillus flavus, and this is Aspergillus niger. There 30 and 200 potency was more effective. Here against flavus, 50M and 200 potencies have given better response. So our drugs are, our potencies are a drug in themselves. This is just to prove that our drugs are not only effective against fungi, but they are quite different in their action, which can be shown objectively through these experiments. Here you will see ketoconazole has given the best response and 4.3%, and Thuja 50M has given 52.17. So and the other experiment was done with the rectified spirit, that is control, in which there is no inhibition against Aspergillus niger. So this work was again republished in Homeopathic Heritage under the title In Vitro Activity of Thuja against Human Pathogenic Aspergilli. This was a collaborative study between me and Dr. A.K. Srivastava, who was MSc and PhD in medical mycology. So this work was highly applauded at that time. Here you will see another fungus, Trichophyton mentagrophyte. It causes, ringworm on the skin. It causes hair loss also. It damages nail, leading to onychomycosis. So here you will see that compared to rectified spirit and ketoconazole, Thuja has given a very, very good inhibition in 30 potency, 200 potency, 1M, Thuja occidentalis in 10M and 50M. Here in different potencies, Thuja, it is effective against Trichophyton mentagrophyte, which is very, very resistant fungus, which can only be killed by ketoconazole, which is a highly toxic drug of allopathy. So this slide is another, it's very technical. You see the green one shows inhibition and the red one shows the growth. So here in different potencies of Thuja, we got different results. This is another slide showing effect of Thuja in different potencies on Microsporum species. Microsporum is the fungus which we have taken out from the hair, infected root of the hair. It is a big cause of hair loss. Many people, many clinicians do not know, but in all the cases of hair loss, we got the hair tested and found out Microsporum in about 86% cases. In other remaining cases, Candida and Trichophyton was found. So the basic purpose was of this experiment was to show broad spectrum activity of thuja in different potencies. So on microsporum, you will see compared to control of ketoconazole, you will see 30 thuja in 30, 200 and 1M potency has given better result compared to other potencies. This is another diagrammatic graphical presentation on the activity of ketoconazole has given 78% result, whereas thuja in 1M potency has given 83%. Sometimes you get much better result compared to ketoconazole. So this is very interesting. This is percent growth of inhibition of microsporum RDI recorded on 9th day of post-sporulation. So the basic, I would like to emphasize the basic purpose of this experiment was not to find out any antifungal drug, but to show to the world that homeopathic drugs in potentized form are effective against these pathogens isolated from human being. The technique, the technique of conducting all these experiments was very, commendable. Okay. This work was again published under the title Antimycotic Potential of Thuja against Dermatophytes in vitro studies. It was published in Homeopathy for All in the year 2002. This is the effect of drugs, thuja in different potencies on Carvularia lunata. This is a plant fungus. It affects human being also. In thuja 30 and 200, you see the result is almost equal to ketoconazole. And in other potencies, it is effective, but not as good as thuja 30 and 200. So if you compared with the outcome, so out of various medicines tested, Meserium 200, by the way, showed maximum inhibition to the growth of Candida albicans. Thuja 30-200 were effective against Aspergillus flavus and in 50M against Aspergillus niger. Thuja in different potencies were effective against Trichophyton, while no potency of thuja was found effective against Microsporum. And thuja 30-200 were highly effective against Carvularia lunata, while antifungal potential of thuja 10M51 are almost clear to equal to ketoconazole. So these experiments are enough to show to the world, scientific world, that these drugs are not placebo. These are having very excellent action on plant fungi and animal fungi. Now coming to the second part of this experimental work, in vitro evaluation of impact of prohibited items and allopathic medicines on the efficacy of homeopathic medicine. The big question before all homeopathic practitioner is whether different edible items have any activity or impact on the effect of homeopathic medicines. The question is, the answer is not yes or no. Nobody can say to the world that what kind of edible item can inhibit the effect of homeopathic drugs unless some experiment is conducted like this. So the basic purpose was, the research question is, in earlier days and even today, homeopathic physicians usually restrict a number of edible items, beverages, and allopathic medicine that they should not be taken side by side with homeopathic medication. So now the question is, do these forbidden items and allopathic medicine nullify the action of homeopathic medicine? Yes or no? Nobody has any answer. Or could any, by any means, such phenomena, if any, be recorded? Just to convince ourselves and to the scientists. So experiment was designed and in vitro experiment was planned to demonstrate the impact of forbidden edible items, beverages like tea and coffee, and number of allopathic medicine on the action of homeopathic drugs in experimental model. Because it can only be shown in experiment, not in vivo or on the patient. Because if you give a drug to the person, suppose you have given lycopodium to one person, and to other person you have given lycopodium, and you have allowed the patient to take onion, garlic, etc. So if drug doesn't work, then how can you blame onion and garlic? Maybe lycopodium, it was not a lycopodium case, or the potency of lycopodium was not appropriate with the patient. So how can you prove that drug has been inactivated by these items? It can only be shown by experiment, which we designed and shown to the world, that I'll show you the results. Now, the objective was to demonstrate the impact of various forbidden edible items and beverages like garlic, onion, cardamom, clove, caraway, ginger, fenugreek, black pepper, asafoetida, red chili, green chili, turmeric, lemon, camphor, and beverages like tea and coffee, and allopathic medicine on the effect of homeopathic drugs in experimental. These are the items which are restricted by homeopathic physician during treatment. So a preliminary effort was made to observe the impact of these items on the inhibition in the growth of pathogenic fungi by homeopathic medicine in experimental model. This slide will depict you, usually all homeopathic physicians, almost all, prohibit these items by the patient not to be taken, like garlic, onion, cardamom, ilachi, clove, long, caraway, jeera, ginger, fenugreek, black pepper, asafoetida, red chili, green chili, turmeric, lemon, camphor, oh my God, camphor, tea, coffee. I don't think any homeopathic doctor is there who do not restrict one or all these items during homeopathic treatment. Now, we have not found any answer in yes or no in the last 225 years, ever since time of Hahnemann. So this was a big question in front of us. Since we developed a research model, so we thought of conducting an experiment to show the impact of these items on homeopathic medicine. Now, here we have taken Candida albicans. Source was fungal isolate from the lesion of the patient. The control, distilled water, vehicle, ketoconazole, and quantity was drug was decided as per MIC. Experimental protocol was DISC method, and zone of inhibition was recorded. Now you see, first of all, we conducted whether there is any activity of these various items on the fungus Candida directly. So here you will see in the slide number 2 or number 1, this Candida, it is treated with Candida, and Meserium 200 has been given on this experiment. It has given a zone of inhibition that shows effect of Meserium. And the second is rectified spirit, it has no effect. Then garlic, cardamom, clove, all these items have been tested on Candida directly, which have no impact on the Candida albicans. So this is one proof that these items do not work on Candida itself. Now, the second question is here. You will see number one is Meserium 200 has given 38.37 millimeter inhibition, whereas all the drugs have, items have not given any zone of inhibition. Now, this interesting is this. It shows impact of forbidden items admixed with Meserium. Now, Meserium on the Candida albicans growth compared to Meserium 200. So here in slide number 1, it is treated with Meserium 200. It is effective against Candida. Now second slide is rectified spirit, that is it has no impact. And now if you compare in the slide number 3, it is mixed, Meserium 200 is mixed with garlic, both given together. Then in other slide, mixed with onion, then Meserium 200 mixed with cardamom, Meserium 200 mixed with clove. They both were mixed. Then they were incubated in under sterile condition and planted on the culture plate. So now if you compare Meserium 200 alone and if you compare zone of inhibition of Meserium 200 treated with these items, you will see almost equal zone of inhibition compared to Meserium, meaning thereby these items, whatever edible items, have not been able to nullify the action of Meserium 200. This is the basic purpose of this experiment, and this is enough to convince all of us that any of these items, including camphor, lemon, turmeric, green chili, red chili, asafoetida, have no impact whatever, even a single % impact on the activity of Meserium. Zone of inhibition is almost equal to the treated one and mixed with Meserium 200. So this is enough to convince any person, any even non-scientific person, that our drugs are quite potent and they cannot be neutralized by these edible items. Here, if you compare Meserium 238.37 and almost equal zone of inhibition is given by the slides in which the drug was mixed with these edible items. So this was very, very interesting. It was very difficult to publish, but unless it is published, it cannot be recognized. So fortunately, by the grace of God and Hahnemann, this paper was again published in peer-reviewed Indian Journal for Research in Homeopathy under the title Impact of Forbidden Food Steps on the Efficacy of Homeopathic Medicine and In Vitro Evaluation. This was published recently in April-June issue of 2018, issue of IJRH. And here you will see Meserium, number 1 is Meserium 200. The other 2 slides, culture plates are treated with tea, coffee. Tea and coffee itself have no impact on Candida. So Meserium has given 37 millimeter zone of inhibition. Rectified spirit has given only 12. Tea has given 12 millimeter. Coffee has given 12 millimeter. And lo and behold, you see in this slide, in this experiment, you will see number 1 plate is treated with Meserium 200 alone. And that number 2 is treated with Meserium 200 mixed with tea together. And the plate number 3 treated with Meserium 200 mixed with coffee, sterilized coffee, of course, sterilized tea. Otherwise, secondary infection would come in the plates. So this experiment was done in totally sterile conditions in a culture room, which we developed in our laboratory. And you will be I mean, this is very shocking and surprising that raw coffee and raw tea have not even antidoted effect of Meserium 200 even to a iota, because Meserium 200 alone has given 37 millimeter inhibition. Meserium 200 mixed with tea has given 34, and Meserium 200 plus coffee has given 37 millimeter zone of inhibition. It is almost equal. So meaning thereby conclusion is that these items, coffee and tea, cannot antidote our drugs, even taken if taken together. This again work was published in Advancement of in Homeopathic Research. This is a journal published from Noida under the title In Vitro Evaluation of Impact of Forbidden Items on the Efficacy of Homeopathic Medicine. The co-authors were myself, Dr. A.K. Srivastav, Dr. Naveen Gupta, and Gaurang Gupta, Sunil Mishra. This was published in February-April 2017 issue. These journals are all available, maybe available in the net also. Those who want to refer to can go for that. And here, usually we believe that homeopathic drugs cannot be taken with allopathy. If allopathic drugs are given, then our action of our drug is antidoted. So first of all, we have seen as to what is the impact of prednisolone, ibuprofen, paracetamol, Atenol, and this is anti-diabetic drug, and enalapril maleate on the candida itself. So you will see, compared to ketoconazole and Meserium, these drugs have not given a 1% inhibition of fungus candida. Usually what you see, allopathic doctor prescribe prednisolone in fungal infection, but if you see in the slide number 4, prednisolone has not a single % impact antifungal activity on candida. And then why this medicine is given? Why? I tell you, this is a immunosuppressive drug. It suppresses your immune system. It doesn't kill the fungus directly. Whereas our Meserium 200 is inhibiting the fungus directly, like ketoconazole, which is a known anti-fungal drug. So here you will see there is no impact. If you compare this impact of allopathic medicine alone on the growth, yeah. Impact of allopathic medicine as mixed with Meserium. So here what we did during experiment, Meserium 200 was given like in the plate number 2, and number 1 is treated with ketoconazole. See, the zone of inhibition is equal. Now comes the mixing of allopathic drug with Meserium 200. So the third plate is treated with Meserium 200 with ketoconazole. Here impact is almost the same. And in other plates, Meserium 200 mixed with the prednisolone, Meserium 200 mixed with ibuprofen, then paracetamol, then atenolol, then anti-diabetic drug, and Meserium 200 mixed with enalapril. If you compare, if you have a look on all the culture plates treated with Meserium and ketoconazole alone, it has given 40 millimeter zone of inhibition. If you compare others, they have given 39, 38, 37, 38, 37, 37 millimeter, almost equal to the zone of inhibition produced by Meserium 200, meaning thereby that these allopathic drugs have not been able to antidote effect of Meserium 200, which is a known potent antifungal drug against this. So this is a controlled study by which we can show to the world, to the scientific community, that these drugs are not placebo, but these are biologically active, and they cannot be antidoted by these edible items are allopathic drugs, as is being thought by practicing homeopathic physicians. Here you will see number 1 is Mesareum 200, green one. Then number 2 is Mesareum 200 mixed with ketoconazole. It has given a little bit more inhibition because both these drugs are antifungal. And if you compare Mesareum 200 mixed with prednisolone, then almost equal zone of inhibition is visible, meaning thereby these drugs cannot, they have not been able to antidote activity of Mesareum. This paper was again published in Advancements in Homeopathic Research. This is again a half peer-reviewed journal. It was published in 2018 under the title as is visible in front of you. So now the question is, what is the outcome? So these experiments have all clearly demonstrated that there is no inhibitory effect of various forbidden items, beverages like tea, coffee, and number of allopathic medicines on the effect of homeopathic medicine when administered together. Even if they are given together, because our drugs are in potentized form, they are in nano form, they are in energy form, and allopathic medicine and forbidden items have got physiological impact. These are working in physiological plane, whereas our drugs are working in dynamic plane, so they cannot be antidoted by these forbidden items. So this gives a clear message. So now, almost coming to the conclusion, the results of these in vitro experiment remove the misconception that homeopathic medicines are placebo. First of all, it took for me 40 years of my life to prove, because I decided in the year 1979 and 80, when I was in final year, that I will prove in my lifetime that these drugs are not placebo but biologically active, and that has, by the grace of God, I have been able to prove that. The second point is biological activity of potentized medicine is proved by inhibitory action on the growth of candida in culture plate. It is evident from experiment that there is no interference of forbidden food items like we homeopathic practitioners are advocating, beverages, allopathic medicine on the action of homeopathic medicine. And the last one is role of disease-oriented diet restriction, however, remains undisputed and should therefore never be negated. Meaning thereby, if somebody is diabetic, you have to cut short sugar intake. If somebody is hypertensive, you have to cut down fat and salt, etc. If somebody is having peptic ulcer, you have to avoid taking chilies and spices. If somebody is having colonic ulcer, then you have to avoid painkillers. That's another thing. That's a disease-oriented diet restriction, not homeopathic drug-oriented diet restriction. So with this conclusion, my presentation is coming to an end. Of course, this is a very exhaustive work, and the work has been published in this book, my recent publication, Experimental Homeopathy. This book is a compilation of all my experimental research work, including 16 research papers on viruses, animal fungi, human fungi, and human viruses. So with these words, I would like to come to a closer of this presentation. This book is unique in itself in homeopathic world, that nobody has done in nowhere in the world anybody has conducted this kind of experiments which are able to convince even non-homeopathic doctors, non-homeopathic scientists, and even to lay persons. Any educated person can appreciate that these drugs are not placebo, and moreover, these drugs cannot be antidoted by so many forbidden items as being practiced with the practicing homeopathic physician. Now, this is a table of content. It has got two sections, one on virology in which 6 papers are there included. And in mycology, my work right beginning from 1981 to 2018. So these are the work on anti-mycotic, and that is antifungal activity of homeopathic drugs. And 3 papers, last papers, are having dedicated to the impact of forbidden items, beverages, and allopathic drugs on the impact of. So anybody who is interested to go through this unique masterpiece work, the book can be, it's available now in Amazon also. And coming to an end, I just give you a very, very big thank you, Dr. Srija, Dr. Sweta, and Dr. Kavita, even in her absence, for giving me this good opportunity to present before you whatever work I could be able to do in the last 40 years to prove that homeopathy is science. It's not a myth. It's not a myth. It's not a placebo. So thank you very, very much. Goodbye to all of you.
Thank you so much,
Thank
sir,
you very much.
for this enlightening session. It was really wonderful. And Dr. Shrija, would you like to ask some questions from the viewers?
As of now, there are no questions, ma'am.
We have with us Dr. Hira, and
Yeah.
he has raised his hand. Sir, you can please ask your questions if you have any.
Sir, good evening, and good evening, everybody.
Good evening, sir.
Gupta ji, it was a wonderful experience. Your long enlightenment of 40 years experience you have narrated here. But whatever I have seen, the problem with homeopathic experimentation and research is, like you had taken here all sorts of ringworm and fungal infections. A lot of fungal infections are coming up now, which are resistant to all type of allopathic medications. The problem with homeopathy, sir, I think I'm clear. I'm audible.
Yeah, sure. I'm able to hear you.
Sir, so the problem now with homeopathy is the modalities. People come with a lot of fungal infection, resistant fungal infection, taking all muconazole, all this ketoconazole. And when they are better with warm applications, generally arsenic followed by tuberculum helps them. If they are better with cold, it is mostly tellurium that helps. Okay. Many a times in a resistant case, syphilinum has worked where night aggravation was more and there was a difficulty in modalities.
So you are talking of clinical. This was not a clinical work. This is totally experimental work. The basic mission was to show to the world that this is not placebo. Even nowadays, people are telling homeopathy is placebo. It has no impact. So we are not discussing clinical aspect of this. We can have another session on clinical aspect.
The problem is in vitro, many things have come up. I have worked with earthworms also and worms also, where it has shown the roundworms and pinworms that are there in the body for the children. Our remedies are working. It has shown in vitro. There is no doubt about it with their santonin and all our remedies. The problem here is, one is it is working. There is no doubt about it. When you come as an individualized or clinical situation, the difficulty is how to put that in a research format. One question for you. Second is what you talked about. You have taken only miserium as a remedy for the antidotal action. Now, there is a full chapter on relationship of remedies, antidotes, relationships. Okay, follows well, complementary, supplementary. Ignatia has a very strong action or antidotal action with coffee. So if you give Ignatia and the patient is taking coffee, definitely there will be a retarded effect of Ignatia, or there will be no effect of Ignatia. Onion, Allium cepa have been given and the patient is eating raw onion, there will be an antidotal effect on the patient after taking raw onion. So antidotal effects is seen in general. A rule has been done because we don't know which remedy we are giving when. So remedies that are having strong medicinal effects on people are being used as antidotes, and they are supposed not to take during the time of treatment. This is what a general rule has been done. Otherwise, there is no necessary of putting this relationship of antidote. There are specific antidotes for specific remedies. Ignatia will definitely have a very strong antidote with effect with coffee. Tea in general is not there. So though we have seen that patients sometimes drink coffee, it does not, it retards. जैसे अपन चल रहे हैं और सामने से हवा चल रही तो अपन पहुंचेंगे पर देर से पहुंचेंगे, और चल रहे हैं अपन और पीछे से हवा चल रही तो अपन जल्दी पहुंचेंगे. यही फर्क है एंटीडोट और या इसमें कॉफी या कच्चा कांदा कच्चा लहसुन लेने में. तीसरा जो आपने बताया है कि what was the third situation you talked about? So this is what I feel about the antidotal action. Definitely there is an antidotal action of the remedies because you have taken only miserium. Other remedies, miserium has no antidotal action of any other food ingredients that we take around. So if you go through the chapter of relationship of remedies, you can see very specific antidotal effects of these ingredients. The third, what you talked about was, I forgot that the third part I did not write. So these are few things I would like to bring to our kind consideration about the discussions that you have done. And I would like to learn from you how to do research on a clinical based trial where we have patients, single paper design. A patient of chronic fungal infection is treated with syphilinum. He is better with syphilinum only. How you have to prove that or show that? We have photographs before, after, everything. So difficulty is in a particular situation because you have an individualized remedy and we cannot compromise on that. Even the potencies, as we have shown here, potencies may play a big role. Some will be only sensitive to 30, some will be sensitive to 200, some will be sensitive to 1M. So again, the potency plays an important role in the sensitization of the patient. Or intercurrent remedies also will be needed. Many a times thuja followed by arsenic is there, many a time tuberculum followed by arsenic is there, depending on the modalities. So in general, I would like to learn from you how to do research in clinical way or clinical patients. If you can guide me for that, I will be ready to follow you in that particular aspect, because a lot of people is patients are there where we have to, where different remedies are helped for the same disease, and how to bring that out into a research model is a big question for me. Thank you. No voice, sir, no voice.
Please unmute yourself.
Hello.
Yes, sir. You're audible.
Hi, Dr. Shweta. The basic purpose of this work was to show to the world homeopathic drugs are not placebo. They are not placebo, they are biologically active. I told you in the beginning, my purpose was not to find out any antifungal or antiviral drug, but to show that this is not just a plain water or it's a aqua therapy, but homeopathic potencies have got action. This was the mission of this work. So if we are, of course, I am practicing for the last part. I have published a book on various skin diseases under the title Evidence-Based Homeopathy in Dermatology. That's another issue. Here the issue was to show experimentally that these drugs are not placebo. This is not a plain water, plain alcohol, but it is different from the placebo. That was the mission. So coming to an end, we can have another clinical sessions, and we can discuss so many other things beyond, limit in near future. So thank you very much, Dr. Shiv. Okay.
Sure, sir. Is there any other question, Dr. Shrija?
No, ma'am.
All right. Thank you, Dr. Girish Gupta, sir, for your gracious presence. And we would like to take the privilege to honor your gracious presence and precious time at our KA webinar with a beautiful certificate, kindly accepted from the team of KA Homeopathy Study Group.
Thank you. Thank you. Oh, that's great. That's
Congratulations.
great.
And we would also like to share the book review of your book, Experimental Homeopathy, which is done by Dr. Kavita. Just give me a second.
कौन है?
Kavita Kokle, homeopath from PSA, President and of Kavita Holistic Approach, founder and director of Homeopathy Study Group Proponent. It is a great honor for me to receive this precious book, Experimental Homeopathy, by author Dr. Girish Gupta, MD Homeopathy. Many congratulations, Dr. Girish Gupta, for coming up with this very unique book, a compilation of in vivo and in vitro experimental research papers published in various journals with the black fungus and white fungus, which no one has done so far. This book is beautifully laid out with historical research papers dating back to 1980 and up to date, accurate homeopathic research data folded in every chapter, enhanced with fortified colored images and graphs to make them more interesting. Dr. Gupta proved that without scientific evidence, the existence of homeopathy as dynamic therapeutic medicine is always questioned. This book, Experimental Homeopathy, is broadly divided into two sections, namely virology and mycology. It contains 16 experimental research papers, 6 on antiviral effect of homeopathic drugs against plant and animal viruses, and 7 on antifungal efficacy of these drugs against human pathogenic fungi. Additional 3 papers have been included on the evaluation of inhibitory effect of forbidden food items, beverages, and certain allopathic drugs, if any, on the efficacy of homeopathic medicines and in vitro models. This book is published by Gauran Clinic and Center for Homeopathic Research. To order your book, visit www.gcchr.com/publications. We, the Homeopathy Study Group Propono team, made a book review of Experimental Homeopathy with author Dr. Girish Gupta, available at our YouTube channel, Kavita Kokno. This book provides beacon light in homeopathic research and needed for researchers, homeopathic fraternity, academicians, and millennials. Thank you so much, Dr. Girish Gupta, for gifting this precious book to homeopathic fraternity.
Thank you, sir. And this is not the end, and we have a lot more to learn from you. We will. Is it fine?
Now it is fine, ma'am. Yeah.
So let me announce the upcoming event in August. We have with Dr. Deepak Sharma, and I'm also going to join him for the session that is on decoding natrum sulfuricum. And we have many more renowned homeopaths who are lined up for KA webinars. Today we have with us our KA volunteer Mohammad Muzammil and Dr. Poonam Chablani, and Dr. Deepa also joined us. We want to thank our KA Homeopathy study group entire team for their continuous efforts, selfless support for making KA activities and webinars successful, and the participants for their undivided attention and inspiration. Dr. Shija, would you like to add anything?
Yes, ma'am. For upcoming webinar details, please follow us on social media. You can follow us on Facebook at KA Study Group page. And if at all for any queries, you can email us at kastudygroup@gmail.com, and also follow us on Twitter, Instagram, and in LinkedIn. Name is KA Study Group.
Thank you. Thank you, Dr. Srija, and thank you all of you for joining us today. Have a great day ahead. Thank you. Okay, goodbye. Goodbye, everyone.
Bye, ma'am.
Shall we end?
Yes, ma'am. Let's end.